Let's start the cell cycle with interphase. Interphase consists of gap 1, which includes the cell growing, gap 2, which also includes the cell growing and as it prepares for meiosis, and synthesis, where the DNA is replicated. Let's take a look now. The 4 chromatids in these 2 homologous pairs are duplicated into sister chromatids joined by a centromere. These are called chromosomes. Let's go on to meiosis to see what happens to the cell in its journey in dividing. During prophase 1, chromosomes also coil and become shorter and thicker and visible under the light microscope. Another thing that happens in prophase 1 is a process called crossing over. In crossing over, the homologous chromosomes exchange genes, resulting in a mixture of parental characteristics and offspring, meaning more genetic variability. The 2 homologous pairs overlap. These points of overlapping are called chiasma. After prophase 1, we move on to metaphase 1. In metaphase 1, the chromosomes line up in the middle of the cell, and they are held there by the spindle fibers. After metaphase 1 comes anaphase 1. In anaphase 1, the sister chromatids remain attached, while the meiotic spindle pulls them to opposite poles of the cell. After anaphase 1, we move on to telophase 1 and cytokinesis 1. In telophase 1, a cleavage furrow appears, and by the end of this stage, the parent cell has divided into 2 daughter cells. This separation of the cytoplasm is called cytokinesis. After cytokinesis, we start meiosis 2, which starts with prophase 2. During prophase 2, chromosomes condense, and the nuclear envelope breaks down if needed. The centrosomes move apart, and the spindle forms between them, and the spindle microtubules begin to capture chromosomes. Next is metaphase 2, where the chromosomes line up individually along the metaphase plate, which is located in the center of the cell. Next is anaphase 2. During anaphase 2, the sister chromatids of each chromosome separate and move towards opposite poles. During telophase 2, the chromosomes reach opposite poles, cytokinesis occurs, and the 2 cells produced by meiosis 1 divide to form 4 haploid daughter cells. The 4 cells produced are considered haploid cells because they contain 1 set of each chromosome.